And this is in a review that I believe I put on the schedule. It's the Ashino review. I think I put it in the discovery session yesterday. So if you look at the schedule there, it should be there. It's a really good review about how this was discovered and just some overall ideas. And what this does, it just shows you, can I take this?
Yes.
So it shows you these classes that Diane was talking about, class 1 and class 2. And what we were saying is class 1 is all about multiple proteins with multiple different functions, all working together into a multimeric complex, right? And so this is one kind of system. And Cas9 seems to be kind of like the important protein in those systems. And in class 2, there are 3 different types. Again, you've got classes, you've got types, it gets very confusing. Type 2, class 2, type 2 is what Cas9 is all about. But all of these are multi-domain proteins, single proteins that are the effector proteins, these endonuclease proteins, single proteins with multiple domains. So the domains are where the different functions come in, not the different proteins. And what we're going to do is analyze those domains today to try to understand how this all works. And over here, just to give you an idea, we were talking about bacteria versus archaea and all the different types and classes you can find. And what you will find is that our class 2, type 2, is a small part of bacteria. And I don't think it's even found in archaea. So that's kind of interesting. And so just understand that Cas9 is, yes, it's a thing. But when you talk about biology, it's a very minor thing. The reason, I think we've talked about this already, we'll continue to talk about it, the reason that Cas9, and you'll find also Cas12 and Cas13 are important is because they're one protein, it makes them a lot better to apply this to technological applications, right? It's just, they have different, they're more compact, and based on their different functions, they're good for different things. And so just wanted to give you that overview. All right, any questions on that? And now what I want to do is look at this. So if you can get this out, here's what I'd like to do, because I want you to not get lost, and I want you to be able to take this later and explore it on your own terms. And so there is a JMOL webpage that you can go to after this to rotate this, zoom in, zoom out, explore, and make sense of it. I think if you do it now, it might be too much. So I really want you to focus on your physical model and this, and just listen to the story, make notes on this, and then use that later to try to explore it in 3 dimensions in your JMOL, okay? All right. So first of all, I want to give you a little comparison of some of these models. So you guys have this model, right? I guess I should go like this. This is kind of this is right side up, right? Supposedly. This is your model. What this is, sure, clear off my desk. I have a messy desk. So this model is what has been, in the paper, it's been described as the primed for cleavage structure. So this is right before the cut, okay? This is when it's found its match, it's found the correct sequence, the guide RNA is matched to the DNA that's complementary to it, and it's just ready to cut. But what we are going to do is we are going to back up, and we're going to look at a conformation before that. And what you need to appreciate about Cas, and I like this model somewhat It's got, it's got issues too, but what's nice about it is it gives you the feeling that Cas9 is not rigid like this. It's moving, it's changing conformation depending on what's bound to it and whatever. So you have to appreciate that these domains are moving around and into different conformations that make it active in different ways. Okay, so what we are going to look at right now is actually a model that is in this structure. So this is not bound to DNA yet. This is what they call the surveillance complex, or I saw another word for it. It's the DNA. What is it? The DNA recognition competent conformation. Okay, so it is the one that's looking for the DNA that is complementary to the guide RNA. All right, now, in order to understand it, we want to first, what I did here is I made a model of this that is color-coded. It's close, not perfect, but color-coded to this. This is the primary structure of Cas9. It's 1,368 amino acids long, and it is made up of 2 lobes, a recognition lobe and a nuclease lobe. Okay, you'll see there's a nuclease lobe over here and there's nuclease lobe over here. There's a rec lobe here. It is made up of a helical domain, which is the white part. It's made up of a C-terminal domain, which is this salmon colored stuff at the end here. There is an arginine-rich helical linker that is kind of red here. It's supposed to be purple, but that's this. And then it's got the nuclease domains. And here's that H and H that we were talking about. And for people who are asking, H and H seems to signify the fact that a histidine and an asparagine are critical components of the active site. We'll talk about that a little bit later. And then there's the RUBC, which I don't know what that stands for. I looked it up and all I could get is Reading University Volleyball Club in England. So I think that's not it. But the key about H&H and RUBC is that the reason they're called that is because they are conserved domains that have been found in other proteins and have been studied thoroughly. And so the people know a lot about how these domains work. And what they have found is that there is a histidine 840 and in H and NH, and there is an aspartate 10 in RUBC1 that are invariable. So no matter where you see this, there are always going to be those amino acids in those positions. And that's because they are right in the middle of the active site, the catalytic site. And in fact, they are critical in coordinating. What do you think they may coordinate? They coordinate magnesium. And that is critical to their catalytic activity. So now you know why the magnesium is important, right? So,
and Diane,
yeah.
What did you say the yellow was?
So the yellows are linkers. They're not that important, but they are just there as kind of disordered pieces that don't have a functional reason except that they connect domains. Okay. So now what do you see that it might be a little surprising when you look at these domains? Does anything jump out at you? Just looking at the primary structure. The CTD is the C-terminal domain. So what strikes you about RUBC? So
up into 3.
Yeah. So it's broken up into three positions. And so let's see if we can walk our way through this 3D structure with, maybe you can zoom in a little bit.
Yeah.
So hopefully, and so what I've done on this page is the left side is what we call maybe the front, okay? And then the right side is just 100, it's just flipped over, okay? So I just flipped it over. So you can look on the right side and hopefully you'll see right here is what we would indicate as the N-terminal domain, or N-terminal end, right? The N-terminus, I should say, because it's got that little blue end, right? And if you follow this, and you guys can do this maybe a little bit better when you get in JMOL, but if you follow this, you'll see, and what I did here is I colored it blue, but I colored it light blue, medium blue, and dark blue. And I think in this, you can see it a little bit better. Light blue, medium, and dark blue, okay? So it starts out with light blue. And then it goes to this arginine-rich helical linker. It goes all the way to the other side. And then it starts going through the helical domains, which are helical domain 1, 2, and 3. And we don't really identify those. They are part of the recognition lobe, which we'll talk about what that's important for in a little bit. What happens after the helical domain? If we follow this, now we're coming back to the medium blue of the RUBC. So way down in there. Then we have a little linker. Then we go to the HNH, which is green. Now we have another linker, and then we get to the final RUBC, and then that connects to the C-terminal domain. All right. Now, the reason that I want to show you that is because I'm going to show you where these active sites are, if I can. So right here, yeah, there. Can you see that? Now zoom in a little bit, maybe.
As far as we can
Do we
zoom.
have light?
Yeah.
It seems
Mark, can you open the blinds over there?
a little. We used to have overhead light. Oh, is it too much before?
Yeah.
Okay, so in here, you will see the aspartate. Oh, look at this! We got a little flashlight. Yeah. So I think so. These three. So there is. Let me make sure I get these right. So there is. I got too many notes. But these are it's the D10 position, and then there is a histidine and something else next to it, and those are all part of the active site. Now, what I think is cool about this, oh, here I got it. Okay, so I want you guys to map this on the primary structure. So we know we already have it marked for D10, right? So you see what that is. So now the other two, look at how close they are to each other in that active site. Okay, so the other ones are a glutamic acid 762. So mark that on your primary structure here.
Okay, so hold on a second, Diane, because I can't see both the model and what you're showing.
Okay.
So you're wanting them to mark.
Right,
where
so I
things
want you
are
to take
here?
notes up here. So we got D10 is part of the catalytic site. And then we've got glutamic acid 762 and histidine 983. And tell me what you see when you map those out. Anybody just shout it out.
They're all in the blue.
Yeah,
They're all
there's
in the
one
blue.
each in one blue. Yeah,
I'm sorry, what?
there's one each in each part of the blue. Each part
Right,
of the blue
so,
has one.
so the RUBC is divided up across that whole protein chain, but those important catalytic site amino acids that are right next to each other in the structure are all from completely different parts of that protein that are circling back. So, I mean, I think that is pretty amazing. So when, like Tim was saying, when they do that D10 to alanine substitution to make a nickase, what they're doing is they're changing that center one to an alanine. And alanine they use often because it's like nothing, right? It's got no real, it's a methyl group. So you're just removing any like active side chain. And so that will then inactivate the RUBC, which is what cleaves the non-target strand. And we're going to talk about the mechanism next, but just want you to have a clear idea of that. And then let's see if I can, oh, this is going to get more difficult.
It was 980 what? So 762 and 980 what?
983.
Can you show that, your printout again, just so that people can see the linear map?
Yeah, can we get both of them up on the same, you know, you don't have to show me at all. So just, yeah, just if we can just show the overhead and that.
Just,
Does that work? You guys see it? Okay. So everybody sees that, right? Where those are?
So they're in here.
There and there.
And here.
Yeah. And now I'm gonna try to get you to appreciate, oh, maybe I'll do it this way. And hopefully you guys are seeing these things somewhat on your printout. And if you don't, you'll get to see them later. So here is the histidine that is on the H840 of the H and H domain. And if you look at it from this perspective, basically they're right there. So they're kind of close to each other. But what I will show you is if you look at these two, and we'll go through this mechanism later, but these are the two, this is the central canal, the central channel, and these are the two endonuclease domains. Here, they're right lined up in the center. Whereas here, they're kind of off to the side. And that's because when it's not bound to the right DNA, you don't want it to be active, right? You don't want it chewing up DNA until it's ready. And so because of conformational changes, these endonuclease domains are not in position to cut yet. All right. If that makes sense, I'm going to walk through the process of how this works. Does that make sense? Oh, one other thing I have to tell you about, or a couple more things. So where do I start? Let me think. Okay, so this has to be bound to the dual guide RNA, right? Turns out that without the dual guide RNA, it does not bind DNA. It's very nonspecific. Any binding is very nonspecific. So in order to bind DNA at all, it has to have this guide RNA in it. And what this rec domain does, it basically grabs onto this structure. And I have this is the dual guide RNA that's in here. And I have it printed separately just to appreciate the three-dimensional structure of this RNA. Because when we look at it in foam and whatever, we see it in 2 dimensions, right? And so you have to appreciate that there are, as Tim was talking, there are these stem loops in the tracer. There is this part of the tracer that then is complementary to the repeat part of the repeat spacer piece. And then there is this spacer piece that is going to be complementary to the DNA that it's looking for, right? So, yes.
There was a question about the tetraloop and whether or not that extended from the surface of the Cas9.
Right.
So can you address that?
Yeah, so here's where the tetraloop is right here. And So it does extend slightly outside of that, right? And then the other thing is,
So it would make sense structurally that you could connect those without doing
screwing anything
damage.
else
Yeah,
up, right? And then I think we'll probably talk maybe more about this. We've already talked a little bit, but understand too that first of all, this is a crystallized structure. So some things might not be in it just because they didn't crystallize. And then other things might be truncated because of the way that they were actually truncating them anyway. So we know that they did some truncation of the dual guide RNA to try out different structures. But you'll see that it's got some stem loop here, but it's also missing. And usually there's some more, there's another stem loop out here. There's like 1, 2, and 3. And the third one they cut off trying to simplify things. And later on, it was found that that made it caused a problem. But you can see why the stem loops are, at least some of them are important, because this is binding, Cas9 is binding to this only by structure. It's not sequence specific at all. And so it has to be a certain structure for Cas9 to recognize it and to bind to it. And when it does bind to it, it makes it the DNA recognition competent conformation. And it does that in a couple of different ways. By binding to it, if you look in there, like that maybe, it's a little hard to see because you've got this arginine linker in here. But you know why that arginine linker is probably important? So when that comes in here, it pre-orders that the first 10 to 12 positions of that spacer, which they call the seed sequence. Those first 10 to 12 nucleotides are pre-ordered, which means it's laid out there in a perfect conformation so that DNA can be separated, slapped in there, and checked for complementarity, and then allowed to leave again. So it's all ready to be matched up. But it's interesting that there's an arginine-rich linker there that it kind of lays right on.
Yeah.
And so
You're thinking the double-stranded or the phosphate backbone of the DNA,
right,
which is negative,
right.
would interact with
Right.
arginine.
And I haven't really read that exactly, but I'm thinking that might be part of it. But I do know for sure that the rest of the helical domains definitely interact with that sugar phosphate backbone. So it's all non-specific binding of that thing and laying out whatever sequence that is. It's opening it up and laying it out so that it's ready to be interrogated by DNA. Okay.
You're gonna go
By
over
RNA.
their model as well, right?
Absolutely.
Okay,
Yes.
yeah, 'cause
With
they're
your physical
pulling-
model, we are going to connect it to your physical model, but just hang in there for a second.
And then at some point, can you go over the tracer RNA again that is separate from the model?
So
There were several
Yeah,
questions
yeah.
associated with
And
it.
I'm not
just
sure
to look at
what the best way to hold this is, because it's a little confusing because it wraps around. So let me show you. So starting, if we can think about starting in your foam model with the loopy part, right on the top,
Orange tracer.
right? The orange tracer we're starting with where it kind of loops in. And so that is designated by this, okay? And it's looping, and then we have to see where it goes. So it's got, these are the things that aren't clear in there, right? But this is all, even though it's not orange, so don't worry about the color. What I did here was try to, I think this is group, isn't it? Where I tried to show
yeah.
where, how it was changing, where the direction is. So it's basically going by the color of the rainbow here.
Do you want the color matched model?
Not yet, or
Okay.
color matched model.
Yeah.
Just wait a second.
Mike's larger.
Yeah.
Just wait a second though. No, let's just get through
Yeah,
this
and
and then
just
we can
if you
switch.
can point out, where the
So here,
this and
so
the
yeah,
tetraloop.
so
and
so
the
here we go. So this is still tracer. And remember, it's binding with itself. And what are those called? Those are called stem loops, right? And that is what gives it three-dimensional structure for the Cas to hold on to. So here it is. We're still in the tracer, but then now we're getting to the point where this part here is base pairing with the spacer. And we know it's a spacer because all of a sudden we've got the tetraloop, which is pink here. All right, the pink is tetraloop. And then that is the
So first we'll have the
spacer.
repeat and then the spacer.
You're gonna have the, you're gonna have the, yeah, you're right. You're gonna have the repeat and then the spacer. And this I colored by nucleotide just to show this is part of the spacer. There's actually more spacer here, but this is, and actually I think there's some chopped off, to tell you the truth. But just to show you that that is the part that's being checked, right? That is the sequence that is being, is interrogating DNA. So does that make sense? I know it's a little complicated because of the way that it is three-dimensional, but I think that's what's cool about it, because once you see it in the 3D structure, you understand how Cas then, yeah, let's pull it. All right, let's
Thank you.
quickly do this, but don't get confused by too much of this. So here is another model, which the tracer now is orange. And then he's got,
Tetraloop.
but it's a little bit more than a tetraloop, but the tetraloop is here. I'm not sure what the gray is, but yeah, the tetraloop is just 4 at the end. And then it's going to connect to the spacer, which, or the repeat.
Yeah.
And then the spacer, which is purple in this case. So the blue is the DNA. All right. Let's not get too close. Yeah, so do you have this? Yes, we do. So again, hang in there. We're gonna let you explore at your own pace in the JMOL. But I just want to give you some of the pieces to consider once you're doing that. Okay. So how are we doing on time? All right. Now what I want to do is talk about how this happens. Okay, we know the pieces. The only piece I didn't tell you yet is these 2 amino acids right here. These are arginines. So they are, and you can, so to mark these down, so where are those? I thought I had them on here. Oh yes, arginine 1333 and arginine 1335. So if you want to put that, it's in the C-terminal domain, and it is.
Oh, I didn't actually share. I was trying to point it out.
It's okay. Yeah, C-terminal domain, and guess what those are for? I'm not going to tell you. I'm going to let you tell me. 2 arginines right next to each other. Think about what you've learned so far. I know somebody's got it. What's the most important thing about binding DNA? What's the first thing about binding the DNA?
From the paper, well, we didn't talk about it.
What we didn't talk about. What we made sure not to talk about too much. It is where the NGG,
Aren't
the
they
GG,
positive?
right? Yes, they are.
So they would interact with the negative phosphates, right?
Right. They interact actually with the PAM site. The
Oh.
G's, the NGG, the G's are going to bind to those. All right. Okay. So what binding, I told you before that binding the guide RNA prepares the conformation to be able to recognize DNA. So it does that by pre-ordering the seed sequence, something I didn't identify. The seed sequence, the first 10 or 12 nucleotides in the spacer are the most important for recognition, for cutting. If those are not identical, if they aren't exactly complementary to the DNA, you're not going to get a cut. If they are identical and the other ones after that aren't, often you'll still get a cut, because it really cares about the seed sequence more than it starts zipping it together. And then once it's happy, then it'll cut, sometimes even if the rest of the sequence, the rest of the spacer sequence does not compute. So anyway, so that's aligned, ready to interrogate. And then also these arginines are put into place so that they are in the proper position as well. That conformation is now ready to go. So now it is this, right? It is out, ready to search for the DNA. And you'll see here that there is this central channel. See, we don't often see it without DNA in it, but there's this central channel. And then there's also a channel back here that kind of goes in between the nuclease domains. Central channel and outside, external channel. So here's what happens. One other thing, keep adding things. See the gray? You should see this on your thing. The gray right here, that's something called the phosphate lock group. Okay, here's what happens. DNA, this comes floating around and it's going to bump into DNA. It's going to bump around, and anytime it finds GGs, NGG, and GG, it's going to bind those Gs or those arginines. And what happens when it does that, if you look at this, you can see the GGs and the phosphate lock group are like right across from each other, right? So the DNA binds right here. What happens is that phosphate lock group grabs the phosphate backbone of the opposite strand to where these the PAM binding site, right? So it binds the one side, and then the phosphate lock group grabs the other side, and it induces a kink. And by doing that kink in an ATP independent manner, it melts the two strands. It allows the two strands to separate, and it kinks them so that they're 90 degrees to each other. The target strand is going to be plastered up against that seed sequence, right, in the guide RNA. And the other one is going to be passed around to the outside so that they're separated, so that they can't come back together again. That is called an R loop. If you ever see that, it's a DNA RNA hybrid with an empty strand. The DNA is separated from that hybrid. So it checks. And if the first ones don't match up, it immediately lets go. If the first couple match up, then it's going to go further and further and further, and anneal those together. And if it matches up far enough, there's a conformational change that causes the HNC. So here, where we can look at this, it causes this HNH, I should say, HNH to move over into position so that it is able then to cleave the target strand, the one that has been annealed to the spacer, and then the RUBC can come in and cleave the non-target strand that's circling around the outside.